Thank you. It's wonderful to be here, and I've really enjoyed the session so far. We're going to change direction a little bit, and I just want you to clear your mind of, first of all, I want to say glaucoma, right now, everything that we do for glaucoma is lowering pressure, and so that's really our current target, even though we would love to have other methods. So couldn't agree more with the previous speakers. We know about glaucoma, it's progressive, there's vision loss, and this is the only way that we know how to treat it right now. We used a lot of different medications, a few were just raised, and we had a few surgical techniques that were sort of our bread and butter of glaucoma care, and will likely remain so based on data, including the recent Canadian report that came out on MIGS. So you might want to just have a look at that. But just very quickly, I usually have about 20 minutes to give this sort of a talk. So I'm going to, it'll be a bit of a wild ride here. So production, we know, is here. Aqueous leaves through the trabecular meshwork, and we also know that the uveoscleral pathway is involved. But what keeps transparent the aqueous? Why do you have such concentration of protein in the cellular body and the aqueous is like 1%? Well, the lymphatic circulation is a good bet. It drains fluid, it removes waste, and it defends immune function. But in the textbook, it says there are no lymphatics in hair, there are none in nails, and there are none in the eye. Well, that just doesn't make sense. So the question is, could there in fact be lymphatics inside the eye? And I'm hoping by the end of this session, you'll all look at things very differently. So let's talk about lymphatic circulation. This is a major circulation that we never talk about. 3 liters every day flows through man. It resides in our tissue spaces. It depends on high pressure to low pressure zones between arteries and veins, where you have collections of these small capillaries, these lymphatics, and 5 minutes. Well, we'll see how far we go. I hope you'll give me a few more minutes at the very end to complete this. Thank you. Thank you. So these lymphatic capillaries are blind ends that suck the fluid from within our interstitial spaces. So an illustration of this is, here's a blind end capillary sitting in of the lymphatic sitting inside. The fluid is drawn in through it, and the capillary end draws it in, and it enters by one-way valves. The valves only allow fluid to go through one way, so there's no black flow. Eventually, this propels the lymph to move through the larger lymph vessels, through the nodes, into the neck, where they eventually drain back into the venous circulation at the base of the neck. So that's our circulation, sort of lymphatic circulation 101. So back to the question, are there lymphatics in the eye? So we were very interested in this question, and here we looked at human postmortem tissue. This is the ciliary body, and here you can see clearly there's a distinct lymphatic vessel in green and blood vessel. The lymphatic is with potoplanin. This is a specific marker that they use in breast surgery pathology to determine if there's lymphatic spread. And this was also identified by EM. Our question was, well, if they're there, is there flow through them? So through that, we had to use an in vivo model. We used the sheep. The eye is very similar in size to the human. We did radioactive studies. In short, the radioactive tracer was found within 15 minutes of sacrifice in a lymphatic vessel laden with LVE1. So we were confident about this, and this was published, and this was the first publication of the identification. We called it the UV lymphatic pathway. So now that we know that it exists and that it flows, can we quantify it? So we used an approach that a colleague in the past used in order to try to quantify and understand the lymphatic, what was in the leukemic patients. And so it was by gravity drip. And you can see here what we did. We did the same thing. By gravity drip, the sheep was placed. We collected the substance, the lymph from the cervical and thoracic ducts, and we looked at the composition, and there it was, and a percent was lymph. So this was also published. And of course, the sheep is a huge animal. It's big, it's expensive. There was no way we could continue studies with that, so we moved on and described similar findings in the mouse, and that's how we continue to study this. But let's just turn our attention back to the eye. So it looks like there's a lymphatic circulation in the ciliary body, and it's quite rich, and probably some of the movement through the lymphatic circulation would be through the ciliary body contraction. And so lymphatic drainage is really a third outflow pathway. And again, we refer to this as the uveal lymphatic pathway because it exists in the uvea, but it also takes into consideration that this is composed of lymphatic. You can see there's a very rich network with protoplanin. So I think, we all know that now that there are lymphatics in the eye. So, and this is a reference that I would encourage you to go to. It has all of the previous work. We were, of course, very interested, now that we know that can it be manipulated? I mentioned the mouse model. And so in that model, we took mice and used hyperspectral imaging. They were put to sleep, imaged in hyperspectral imaging, and then allowed to wake up, run around, and then sequentially done it. But in some mice, we pre-treated with latanoprost, and some mice we didn't. And latanoprost, as you know, is a widely available drug, most widely. And so what we found was that the speed by which the lymph moved from the eye to the node was dramatically improved or increased in the group that was treated, so in a nutshell. And so we're very excited to be able to share a technology and an assay that we've developed now that was just recently published, in which we can now measure active lymphatic drainage in the live mouse by non-invasive near-infrared imaging. And so I would encourage you to, if you're interested in this area, to please go and read that. But the implications of this go beyond glaucoma, as you can imagine. If we have lymphatics, it helps us understand aqueous dynamics, glaucoma, immune surveillance, cancer spread, inflammatory eye disease, and even space flight. So now I want to just turn, I was also given the title Glymphatics, so it's not a typo. There's lymphatics and there's glymphatics. So if we look at the back of, if we remember that we have a circulation called the cerebrospinal fluid, which surrounds the brain to provide buoyancy, nutrient delivery, and clearance. And this fluid is also surrounding the optic nerve. So we used mice, and we performed some studies and injected substance into the cisterna magna, which makes its way into the circulation so we can visualize it with some dyes. And we know that both the eye and the CNS, the fluids, are in very close contact with one another. And our question was, what is that relationship? We know that the CSF flows around in the subarachnoid space to the optic nerve here. And there's a lot of interest in the relationship between CSF and glaucoma in relation to the optic nerve. But how much do we really know? And so when we did these studies, I want to just illustrate what we did. So we injected into the cisterna magna, the dye moved through into the circulation, up through the optic nerve, and this is what we found. So we expect that we would see the dye lining the nerve. We did not expect to see it inside. So inside, so when you inject into the CSF, we saw it enter the nerve inside, and so we said, well, could this be the glymphatic pathway? The glymphatic pathway should be thought of as a major clearance system for toxins in the brain. It was recently described about 5 years ago, and we thought maybe the glymphatic system exists in the optic nerve and we didn't know about it. But in order to determine it's a glymphatic system, you must, here's the CSF, and you can see high power. The CSF is coming around the nerve, and this is a blood vessel entering into the very substance of the nerve parenchyma. And it depends on glial cells and astrocytes, which we showed in our publications, and I'll move forward with that. So this is our this publication showed that there was evidence for movement of fluid around the space into the nerve via a glymphatic pathway. So this is identification. So now you know its glymphatic pathway exists. There's this toxin clearing system that exists. And in fact, this is the way it works. So instead of just around the nerve, it enters the nerve. And you hear it comes through the arachnoid space and lines the blood vessels, and that's how it's getting in. But we were thinking, well, what happens in glaucoma? So we used the DBA model of glaucoma, did the same series of experiments, and this is what we saw. In glaucoma, we didn't see anything inside the nerve. And so this is what we're seeing. And so this was just published in December, so I'm very happy to be sharing it here with you, but there is reduced cerebrospinal fluid inflow into the optic nerve in glaucoma. And I think the take-home message is we've focused on tissue a lot. It's really important that we remember that our tissues are bathed in fluid, that there's inflow, but it means there's outflow, that waste removal from tissues is probably very important. There is a third outflow pathway that might be very relevant. And you can imagine that if you have a waste removal system that is faulty, things may not survive as nicely. And so we hope that this will inspire new hope in the field and new investigations so that we can really someday find a better way for the disease. And so when the next time that you're looking at an optic nerve, remember that probably there's probably more attention to the tissue, the fluids around is just as important. And thank you very much.